Antiviral properties of sunlight. These are notes for February 9, 2026. In this YouTube, we will briefly discuss the use of a portion of the solar spectrum, red light and near-infrared light, for the treatment of viral infections, to prevent viral infections, to treat ongoing viral infections, and to treat people who are recovering from viral infections. What is already known? It is known that sunlight, particularly near-infrared light, will stimulate the production of melatonin within cells. Melatonin will then upregulate the anti-inflammatory antioxidant transcription factor known as NRF2, NRF2. NRF2 orchestrates a set of pathways which reduce the impact of oxidative stress. It is an adaptive mechanism to deal with reactive oxygen species in cells. It is also well known that sunlight, the ultraviolet components of sunlight, stimulate the formation of vitamin D3 in human skin. That compound, vitamin D3, is transported to the liver where it is converted to 25-hydroxy vitamin D3, abbreviated 25-vitamin D3. This liver metabolite is then carried to the kidney and inside the proximal tubules, the epithelial cells of these proximal tubules, primarily within the mitochondria of these cells, 25-vitamin D3 is hydroxylated at the 1 carbon position to form the molecule 1,25-dihydroxy vitamin D3, also known as calcitriol. Calcifediol 25 vitamin D3 and calcitriol 125 vitamin D3 are potent agonists of the vitamin D receptor. So these liver and kidney metabolites of vitamin D3 stimulate vitamin D receptors throughout the body. In doing so, they upregulate the expression of NRF2, the master anti-inflammatory antioxidant transcription factor inside cells. The upregulation of NRF2, once again, serves as an adaptive mechanism to suppress the damaging effects of oxidative stress, particularly reactive oxygen species. Now, condensing this information into an information vector, 125D is an abbreviation for 125 vitamin D3. This could be calcitriol, 125 vitamin D3, or a synthetic analog of calcitriol, paracalcitol, 125 vitamin D2, which is the preferred therapeutic for the treatment of chronic kidney disease patients. On this channel, we have been emphasizing that paracalcitol could potentially be repurposed to stimulate vitamin D receptors in contexts of viral infections and other conditions where there is oxidative stress, such as autoimmune disorders. By doing so, you can stimulate the upregulation of NRF2, which in turn stimulates the production, the upregulation of GPX4, glutathione peroxidase 4, a key enzyme, an antioxidant enzyme, which uses glutathione to reduce reactive oxygen species as well as peroxidated lipids, which are the key upstream trigger for the type of programmed cell death known as ferroptosis. And ferroptosis in recent years has been recognized as a key component of many human disorders and urgent care conditions like sepsis. Ferroptosis is also producing neurodegeneration and Alzheimer's disease, Parkinson's disease, ALS. Ferroptosis is also a key component of the pathogenesis occurring during viral infections, such as COVID infections. So stimulating these pathways, either with sunlight or supplementation with active vitamin D hormones, and stimulating the production of melatonin with near-infrared light These are new approaches to suppress the oxidative stress associated with viral infections and to help individuals who have been struck by viral infections to recover and regenerate tissues. On the right is a schematic diagram, a scheme summarizing the inhibitory effect of lipoic acid on a virus known as the viral hemorrhagic septicemia virus, VHSV. And this suppression of VHSV replication via the activation of antiviral genes and suppression of this virus's induced oxidative stress. This occurs through two limbs. You see the structure of lipoic acid, not seeing the receptor and transduction pathways, but the results. Inside the cell, you see a red box, oxidative stress. Lipoic acid, indirectly by stimulating pathways, including the upregulation of NRF2, stimulates the suppression of oxidative stress. How can NRF2 suppress oxidative stress? One way is NRF2 upregulates GPX4. And by reducing reactive oxygen species and peroxidated lipids, it suppresses a downstream consequence of that oxidative stress, ferroptosis. Oxidative stress is a consequence of the viral infection. Lipoic acid, in tandem with this antioxidative pathway, also stimulates the upregulation of antiviral genes, including viperin. We're going to concentrate on this protein in this YouTube. From here forward, we're moving away from discussing the viral hemorrhagic septicemia virus, and now we are going to be talking about CoV-2 and COVID. First, a review from 2025. Viperin, a multifunctional protein in antiviral immunity and disease pathogenesis. And a news report from February 2024, Murdoch University News in Australia. COVID-19 researchers discover hidden natural immune defense pathway. Professor Nicholson, the ANPC's director, said that they detected 10 new compounds made by the body in response to the SARS-CoV-2 virus. Sidebar comment. These are metabolites being generated by the enzymatic actions of viperin. The discovery of the extended viperin pathway, XVIP, has greatly extended the knowledge of the viperin antiviral pathway, part of the innate immune system that reacts to most viral infections. Viperin is short for virus inhibitory protein, endoplasmic reticulum associated interferon inducible. That is the extended term for the protein, and it is abbreviated viperin. Viperin is a multifunctional interferon inducible protein that regulates virus replication. In other words, it suppresses viral replication. When a virus attacks the body, it triggers an interferon response, and viperin is believed to suppress viral replication. Now, Figure 2 is coming from the review in the upper left. The inducible expression pathway of viperin. After viral infection, PAMPs, pathogen-associated molecular patterns, PRRs and RLR, are activated, which in turn activates the phosphorylation of transcription factors, nuclear factor kappa B, and interferon regulatory factors R, IRF3, and IRF7, and induces the production of type 1 interferons. Interferons can bind to type 1 interferon receptors on the cell surface in a paracrine and autocrine fashion. means they can stimulate themselves. A cell can secrete the ligand when it binds to its own receptor, stimulating that cell. In addition, the secreted factors can activate receptors on neighboring cells. That's the paracrine signaling. And interferons are key signaling molecules which play these roles. Now back to the news report. It is very unusual to discover significant new human biochemical pathways, especially ones that might have major medical significance, Professor Nicholson said. Professor Vist said that the 10 ex-VIP compounds detected, 9 were new to human biology knowledge. These compounds are only produced during active viral infection, and they have very short half-lives. So if we find them in the blood or urine of a patient, then that patient is likely to be infectious and needs to isolate. The ex-VIP metabolites, these are the metabolites of Viperin, its enzymatic activity. So these metabolites, which are generated by the function of Viperin, represent previously unknown biomarkers of infection and infectiousness and may be highly relevant to rapid diagnosis in acutely infected hospitalized patients. Fascinatingly, from a chemical viewpoint, these compounds look like antiviral drugs, such as remdesivir, and are highly similar to some compounds that are already marketed by drug companies as antiviral agents. This ex-VIP opens up the possibility of future studies aimed at designing new antiviral drugs. Of particular note, according to Professor Nicholson, is that human biology contains basic elements of antiviral drug synthesis, and that this has been helping to protect humans since before human medicine existed. Professor Wist said that compounds were detected in both urine and plasma, and a simple non-invasive urine test could also be developed for rapid detection. In fact, we have already demonstrated the proof of principle that we could deploy a rapid urine test for diagnosing active viral infections that could be of considerable medical significance. Here is the Viperin primary structure. There is an amphipathic helix. In the center, there is the radical SAM domain, and at the carboxy terminus, this is where antiviral activity is located. Some background on this sequence. The primary structure of Viperin. The Viperin protein is composed of 361 amino acids and has 3 distinct regions in its primary structure. An amino terminus, which varies in length and sequence with species, a central conserved region, and a highly conserved carboxy terminus. The tertiary structure of Viperin is a bilayer structure containing both alpha helices and beta sheets. The carboxy terminal region of Viperin is also highly conserved and has been demonstrated to be critical for its ability to limit the replication of a number of viruses. Now, down below is a schematic diagram from the paper being discussed in the news report from Murdoch University. Before discussing this figure, it's useful to read the abstract of the paper. We present compelling evidence for the existence of an extended innate Viperin-dependent pathway, which provides crucial evidence for an adaptive response to viral agents such as SARS-CoV-2. We show the in vivo biosynthesis of a family of novel endogenous cytosine metabolites with potential antiviral activities. Two-dimensional nuclear magnetic resonance spectroscopy revealed a characteristic spin-spin motif, indicating the presence of an extended panel of urinary metabolites during the acute viral replication phase. Mass spectrometry additionally enabled the characterization and quantification of the most abundant serum metabolites, showing the potential diagnostic value of the compounds for viral infections. In total, we unveiled 10 nucleoside, cytosine and uracil-based analog structures, 8 of which were previously unknown in humans, allowing us to propose a new extended Viperin pathway for the innate production of antiviral compounds. The molecular structures of the nucleoside analogs and their correlation with an array of serum cytokines, including interferon alpha-2, interferon gamma, and interleukin-10, suggest an association with the Viperin enzyme, contributing to an ancient endogenous innate immune defense mechanism against viral infection. The Viperin gene family extends beyond 3 billion years. So in the bottom part of the slide, you see individuals with active viral infections contributing urine samples. These are then passed through mass spec and NMR for rapid analysis. And then comparing individuals with no active viral infection, they have no urinary signals of these Viperin nucleoside metabolites. Now, when I say Viperin nucleoside metabolites, Viperin is acting on cytosine and uracil-based molecules generating these metabolites, and these are only found during active viral infections. And over on the left, you can see a figure from the paper. So these are NMR signals, and this is the family of nucleoside metabolites that have been generated through the enzymatic action of Viperin, an enzyme. Mammalian Viperin has also been shown to catalyze the formation of 3'-deoxy 3'-4' dihydrouridine triphosphate, DDH-UTP. This is derived from uridine triphosphate, UTP, albeit having an in vitro substrate preference for CTP over UTP of approximately 1,000-fold under physiological conditions. This suggests that these viral inhibitory mechanisms are ancient. The conservation of functional Viperin homologs across 3 domains and numerous bacterial phyla allows a conservative estimate of greater than 3.4 billion years, and subsequently have been functionally tuned by evolution. The presence of antiviral nucleosides in human bodily fluids points to an innate immune capability that is relevant to our understanding of SARS-CoV-2 and other viral infections in humans. An understanding of the immunometabolic processes related to these nucleosides is essential to allow their assessment as diagnostic markers for viral infections. In order to evaluate DDH-CTP as a viral biomarker and to further understand its potential role in the metabolic response to acute SARS-CoV-2 infection, we applied a multi-level spectroscopic approach utilizing NMR spectroscopic and mass spectroscopic methods to ascertain whether the antiviral DDH-CTP is measurable in urine. We detected 10 pyrimidine nucleoside derivatives in human urine, of which 8 of them, to our knowledge, are previously unreported. This indicates a more profound metabolic and evolutionary adaptation to viral infection than has previously been suspected. I would like to connect this paper with a YouTube by Roger Schwelt on the MedCram channel, Flu Deaths Predictable Every Year, and a URL to link to that YouTube is in the upper left. So influenza deaths are graphed here per week in the United States, year by year. And within 1 to 3 weeks after the winter solstice, there is a peak in influenza death every year, the magnitude of which can vary, but the periodicity of this is very strong. Professor Schwelt examines in this YouTube various aspects of the solar year which could be driving these viral infections, ruling out temperature and humidity or social aggregations, because the same type of periodic surges in flu deaths are 6 months out of phase in the southern hemisphere at latitudes that are far from the equator. In other words, where there is little sunlight. High latitude conditions produce not only peaks in influenza deaths, but there are also peaks in many forms of mortality, heart disease. And these peaks are actually larger than the influenza deaths. So the combined influenza and pneumonia deaths are shown at the bottom, highlighted in red. So what is causing this? This paper, Ultraviolet A Radiation and COVID-19 Deaths in the United States, with replication studies in England and Italy. These researchers found that there was a strong correlation between the amount of sunlight measured as the amount of UVA radiation. A lack of UVA, a lack of sunlight, correlated with increased COVID-19 deaths. And this occurred in the US, England, and Italy. Very highly statistically significant. The takeaway conclusion: it suggests that optimizing sun exposure may be a possible public health intervention, given that the effect appears independent of a vitamin D pathway. It suggests possible new COVID-19 therapies. So I would like to tag on this concept. This study, as well as others, illustrate that sunlight has antiviral properties besides vitamin D. What are the components of sunlight that are doing this? And this brings us back to red light and near-infrared light. And what is already known is that sunlight, particularly near-infrared wavelengths, stimulate melatonin. And melatonin upregulates the anti-oxidation, anti-inflammatory transcription factor, NRF2, which suppresses oxidative stress and reactive oxygen species, which are very damaging and can produce cell death by inducing ferroptosis. So I'd like to condense these concepts into a information vector that can be used elsewhere in this YouTube. Melatonin stimulates the upregulation of NRF2, which in turn upregulates GPX4, glutathione peroxidase 4, a key enzyme in reducing reactive oxygen species. GPX4 utilizes glutathione. By utilizing glutathione to reduce reactive oxygen species as well as peroxidated lipids, melatonin, through this cascade of events, upregulating this key anti-oxidation enzyme, is able to suppress ferroptosis. And ferroptosis has now been linked to pathogenesis in many microbial infections, including viral infections. It's also a driver of cell death in neurodegenerative disorders such as Alzheimer's, multiple sclerosis, and ALS. It's also a component of long COVID. So stimulating melatonin with near-infrared light is a therapeutic approach to treating tissues that have been impacted by viruses in particular. In concluding this YouTube, I would like to read from one last slide from the MedCram YouTube, Flu Deaths Predictable Every Year. They're discussing a paper that was published in June 2022, Melatonin Research, a geographical approach to the development of hypotheses relating to COVID-19 death rates, by Scooch, Schwelt et al. Their analysis was limited to countries with more than 50% of the population being overweight. It is well established that age, obesity, or diabetes are key risk factors for COVID mortality. Here, these authors are indicating that latitude and time of year, particularly lack of sunlight, is also a risk factor for COVID mortality. Lack of sunlight, particularly near-infrared wavelengths, will depress melatonin biosynthesis. A lack of melatonin will lead to a drop in NRF2 and likely a drop in GPX4 levels. And with that, the susceptibility of tissues to ferroptosis increases. This type of cell death occurring in the epithelial sheets of the lung and the gut are potential ways that bacterial and viral agents can penetrate into deeper tissues of these organs, leading to sepsis. And sepsis is a common driver of COVID mortality. So, summarizing all of this, sunlight appears to have potent antiviral properties, and utilizing specific portions of the solar spectrum, particularly red and near-infrared light, that therapeutic approach is rapidly gaining adherence in many populations around the world. Besides traveling to sunny regions for winter months, the utilization of red and near-infrared lights, whether it is a focal illumination or whole body illumination, these are potential ways to boost cellular melatonin to drive the production of these anti-inflammatory, antioxidative, antimicrobial pathways. I would like to end this YouTube by relating these concepts to the life of a famous Norwegian composer, Edvard Grieg, who suffered from severe chronic respiratory issues throughout his life, stemming from a serious case of pleurisy, that's inflammation of the lung surface, and tuberculosis, which he contracted at age 17, which caused a collapsed left lung. Despite this, he maintained an active, albeit fragile, career as a composer and pianist until his death at age 64. Grieg would travel from Norway to Italy to gain the light and warmth of that country. Although he loved Norway, he believed the conditions were killing him. He could not go through a Norwegian winter. So certainly by getting the sun of Italy, he was stimulating melatonin, boosting NRF2 and GPX4, suppressing ferroptosis from lingering latent tuberculosis in his right lung. And many people in the 1800s and early 1900s, before antibiotics, had to move to warmer, drier climates with ample amounts of sunshine in order to survive their tuberculosis infections.